Welcome, everybody, to a new year of Fragile Nucleosome. We're excited to keep the seminar series going, um, in no small part thanks to Epicipher for helping sponsor us. But first off, today we have Alan Au from Rob Close's lab at the University of Oxford, and he's got a really exciting story to tell us about that he just pre-printed. Alan got his start doing his bachelor's at King's College London, then a master's at University College London, and now he has moved on to Oxford, where he joined Rob Close's lab to do his PhD, and now he is continuing on as a postdoc to get experiments done and get papers published. And today he's going to tell us about his really cool story with SET1-MLL and how they control transcription independently of their enzymatic function. So, Alan, please take it away.
Okay. Thanks very much for giving me the opportunity to talk about my research here on fragile nucleosome. I'm very pleased to be here. So today I'm going to talk about the work that I've been doing for the past number of years in the Close lab here at Oxford, studying how H3K4 methyltransferases regulate transcription. So our lab is interested in how CpG islands regulate transcription. And so CpG islands are these short stretches of the genome in which you have elevated CpG density, in which the DNA remains free of methylation. And this is in contrast to the rest of the genome, which is pervasively methylated. And we and others have long believed that CpG islands are important gene regulatory elements. And one of the reasons for this is the existence of a zinc finger CXXC domain that many proteins have. And this domain recruits these proteins specifically to CpG islands. And it so happens that many of these proteins are chromatin modifying enzymes. And so we believe that a function of CpG islands is to recruit these chromatin modifying enzymes to CpG islands, where they can modify the surrounding chromatin to create an environment that is either permissive for transcription or repressive for transcription. And furthermore, mammalian gene promoters, over 2/3 of them contain a CpG island. And so we believe that CpG islands have a really critical role in controlling the earliest stages of transcription. And one group of proteins that go to CpG island promoters are the SET1-MLL proteins, which are histone methyltransferases. And they deposit a modification called trimethylation of lysine 4 on histone H3. And this is a very conserved modification that is almost universally found at the 5' end of actively transcribing genes. And it's a modification that is associated with active transcription. So it's usually present at levels that scale with the transcriptional activity of the associated gene. So at a more highly transcribed gene, you will have higher enrichments of H3K4ME3 compared to a more lowly transcribed gene where you have less. And so it's long been hypothesized that this modification must play some important fundamental role in instructing or promoting active transcription. And one of the reasons for this is the existence of protein domains that specifically recognize H3K4ME3. And many proteins that have these so-called reader domains are themselves either part of the transcriptional machinery, such as most notably the TAF3 subunits of TF2D, or are involved in the process of transcription or associated with active transcription. And so this includes chromatin remodelers, histone acetyltransferases, etc. And so a very simple, but actually very intuitive model for how CpG islands can promote active transcription is through the recruitment of SET1-MLL proteins to CpG island promoters, where they deposit H3K4ME3. This then either recruits or stabilizes the binding of effector proteins, which then exerts some function on transcription. But it's actually been very difficult to test this model experimentally because of the sheer number of SET1-MLL proteins that exist. So in mammals, there are at least 6 SET1-MLL proteins grouped into 3 paralogous pairs. So you have the SET1A and SET1B proteins that associate with CpG islands through an interaction with CFP1. You have MLL1 and MLL2, which have their own CXXC domains. And these are the 4 methyltransferases that associate with CpG islands and are thought to be the primary contributors of H3K4ME3. And in addition to this, you have the ML3 and ML4 proteins, which are not CpG island associated, but rather go to enhancers where they deposit mono and dimethylated H3K4. And so having so many metal transferases depositing the same modification makes it very challenging for us to understand whether these histometal transferases actually regulate transcription through the modification that they deposit. And in part, this is because it's been very difficult to understand which complex contributes most to H3K4 ME3. And in doing so, we don't really know whether this modification is important for their transcriptional control or not. And to further complicate matters, we and others have in the past found that these SET1-ML proteins can actually influence transcription independently of the metal transferase activity. And so it's really not well understood whether SET1-ML proteins actually need to deposit H3K4-ME3 to exert their functions on transcription. And so we first sought to understand how SET1-ML proteins shape H3K4-ME3 throughout the genome. And we did this by endogenously tagging the SET1-ML proteins in mouse embryonic stem cells using a Degron tag. And this allows us to very rapidly deplete these proteins upon addition of the D-tag small molecule. And when we did this with SET1A and SET1B, we found a pervasive but very modest reduction in H3K4-ME3. And similarly, when we depleted ML1 and ML2, we found somewhat greater but still very modest reductions in H3K4-ME3. So on their own, these two groups of enzymes do not contribute majorly to H3K4-ME3. It's only when we co-deplete SET1AB and ML1-2 that we start to see major reductions in H3K4-ME3. And this indicates to us that these two groups of metal transferases really work together to sustain H3K4-ME3 levels genome-wide at promoters. But we did note that this reduction was not complete and that there were still substantial amounts of H3K4-ME3 remaining. And we wondered whether ML3 and 4, which are not CpG island associated and are thought primarily to be mono and dimethyl transferases, might have a greater contribution to H3K4-ME3 than we previously understood. And so we generated a cell line in which we could deplete all 6 SET1-ML proteins simultaneously. And when we depleted these proteins for 2 hours, we found somewhat greater reductions in H3K4-ME3 at promoters. But interestingly enough, if you extend the depletion to 8 hours, you actually see a recovery of H3K4-ME3 levels. And so this tells us that there must be a missing metal transferase that was still able to contribute to H3K4-ME3 in the absence of the 6 SET1-MLL proteins. And so we next aimed to identify this mystery metal transferase by mass spectrometry. And initially, when we first started this experiment, we had the intention of convincing ourselves that we had really depleted all SET1-MLL proteins. And we were fortunate in that the SET1-MLL proteins are actually very poor metal transferases on their own. So they need to associate with WDR5, RBBP5, Ashto-like, and DPY30 for optimal metal transferase activity. And these shared subunits we term the WRAD subcomplex. And so we reasoned that if there was a remaining SET1-MLL protein, it would definitely be associated with WRAD. And so we put a flag tag on WDR5 in our cell line where we can deplete all SET1-MLL proteins. And then we performed IP mass spectrometry. And we were fortunate enough to have Inga from Mikhail von Meulen's lab to do this experiment. And she found that after D-tag treatments, SET1A and all the MLL proteins no longer associated with WDR5. But surprisingly, SET1B continued to associate with WDR5. And this was very surprising to us because we thought we had depleted SET1B with our Degron system, and we thought our Western blots showed us that. And when Inga took the SET1B peptides from the mass spectrometry and she mapped them back onto the amino acid sequence of the SET1B protein, she found that these peptides only came from a very specific part of the SET1B protein, which is a C-terminal region of the protein. And this is important for 2 reasons. Number one, our Degron tag was initially placed at the N-terminus. Secondly, this region contains all the domains required for metal transferase activity. And so we thought that there was a C-terminal SET1B isoform that was remaining, and this was contributing substantially to H3K4-ME3. And we confirmed the existence of the C-terminal SET1B isoform using by Western blots. And funnily enough, you can only detect this SET1B isoform with an antibody raised against the C-terminal part of SET1B, but not when you use an antibody raised against the N-terminal region. So this is a C-terminal SET1B isoform. And when we looked at some publicly available data, we found an internal cage-seq peak. So we believe that this represents an internal transcription start site within a downstream instron of the SET1B gene, and that's how this protein is produced. So we now turn our attention to trying to understand how these proteins control transcription. And we used two parallel approaches. So firstly, we generated a cell line where we could deplete all SET1 MLL proteins, including our newly discovered C-terminal SET1B protein. And this would allow us to remove both methyltransferase dependence and methyltransferase independent functions that they exert on transcription. And in parallel, we generated a cell line in which we only inactivated methyltransferase activity by depleting RBBP5 as subunits of WRAD. And in both cell lines, you get big reductions in H3K4ME3. But when you just inactivate methyltransferase activity, you do not prevent the SET1 MLL proteins from associating with their target sites throughout the genome. And so we looked at the nascent transcriptome using TT-seq, and we found that when you deplete SET1 MLL proteins, you have a very large effect on transcription, which over 5,000 genes reduced in transcription. And so this is about a quarter of all genes. And some of these genes were more or less entirely dependent on SET1 MLL proteins for transcription. In contrast, when you just inactivate the methyltransferase activity of SET1 MLL proteins, you have a much smaller effect genome-wide on the nascent transcriptome, with only hundreds of genes that are affected. And most importantly, if you look at genes which are dependent on SET1 MLL proteins for transcription, these genes actually do not require H2K4ME3 for transcription. So you can inactivate methyltransferase activity, K4ME3 will turn over, but as long as the SET1 MLL proteins are still physically associating with the gene promoter, transcription can continue. And so we conclude that SET1 MLL proteins primarily control transcription independently of H3K4ME3. So we next wanted to understand how SET1 MLL complexes control transcription. And we wanted to do this within the context of living cells, in which transcription is a stochastic and pulsatile process, in which multiple polymerases are loaded onto a promoter, and they transcribe as these polymerase convoys that create bursts of transcription that we refer to as on periods. And ultimately, it's the mechanisms that control this behavior that shapes the transcriptional outputs. And so we wanted to know how SET1 MLL proteins control these aspects of transcription. And my colleague Alex took an imaging approach to visualize this in living cells, in which MS2 repeats are engineered into a gene of interest within a cell line expressing GFP fused to MCP. So when the gene is transcribed, MCP GFP binds to these MS2 repeats. That creates a bright fluorescent focus that can be measured, and this can be done for individual cells over a number of hours. And from this, you can extract and quantify the key parameters of transcription. And this includes the number of on periods, their duration, and also their amplitude, which is the number of transcripts produced per on period. And this corresponds to the number of polymerases within each polymerase convoy. And so Alex took a SET1 MLL dependent gene called PANK2, and he imaged this gene over 4 hours. And when he depleted SET1 MLL proteins, he found a very profound reduction in transcription across the entire cell population. And when he quantified the aspects of transcription which were most affected, very strikingly, he found a reduction in the amplitude of the on periods, meaning that the bursts were getting smaller. So you had fewer transcripts produced per burst, meaning that there were fewer polymerases within each convoy. And when he co-depleted SET1 MLL complexes with the restrictor termination complex, he found that he could completely rescue these effects. And we had previously identified SET1 proteins as having a specific role in antagonizing the termination activities of restrictor. And so we believe that in the absence of SET1 proteins, restrictor is able to terminate polymerases within each burst. And importantly, when he depleted RBBP5 and just inactivated methyltransferase activity, he didn't see this effect. So this mechanism of transcriptional control by SET1 MLL proteins by antagonizing restrictor is completely independent of H3K4ME3. And so with these experiments, we conclude that although SET1 MLL complexes shape H3K4ME3 throughout the genome, their primary way that they control transcription is not through H3K4ME3, rather it's by opposing the activities of restrictor. And in the absence of SET1-MLL proteins, restrictor is able to stochastically terminate polymerases within each on period, such that the burst then gets smaller, and that leads to a reduction in the transcriptional output. And with that, I would like to thank all my colleagues in the close lab and our collaborators who contributed to this project. Specifically, I'd like to thank Alex for his work on the imaging experiments. I'd like to thank Inga from Mikhail Vermeulen's lab for the mass spectrometry experiments. I'd like to thank Rob for being a fantastic supervisor. And I would like to acknowledge the Wellcome Trust who funded all our research and supported me through my PhD. And I'd be happy to take any questions that anyone might have.
Great, thank you so much, Alan. Everyone in the audience, you can put questions into the Q&A and I can ask them for you, or if you raise your hand, I can allow you to unmute and ask your own question. I'll start off by asking, when you first showed the reduction and then recovery of K4 trimethylation, I immediately assumed that it was something that wasn't like SET1 that was coming in and methylating, like one of the PRDMs that can act as a methyltransferase there. And so seeing this C-terminal transcript is really fascinating. But did you also investigate whether or not there were non-SET1 proteins that could also be depositing the methylation sites?
Yeah, so initially we assumed that it wasn't a SET1-MLL protein, and we did think of looking at the SMID family of methyltransferases and various other methyltransferases that people have in the past thought might be H3K4 methyltransferases. So there are some methyltransferases like ASH1-like, for example, that people have at various points thought, in addition to being a K36 methyltransferase, could be a K4 methyltransferase. But I think the evidence really came when we started to generate Degron lines, not of the SET1-MLLs, but of the WRAD subunits. And when we made those cell lines, we didn't see this recovery, and we see much bigger depletion of K4 ME3 with longer treatments. And so it was at that point that we thought there must be another SET1-MLL protein that was left.
Yeah. So then we have a question, an anonymous question. Assuming that all MLL SET1 proteins have similar functions, can the transcription phenotype be rescued by a catalytically dead version of SET1?
Yeah, I think this is what we were trying to do with the RBBP5 Degron. I think we would like to, ideally we would use catalytic point mutants, and we actually know which residue we need to mutate within these very highly conserved set domains to inactivate the methyltransferase activity. The problem is that number one, these are very big proteins, and so we want to have a system where we could very quickly switch out the wild type proteins with catalytic point mutants, and that might involve us expressing a long coding sequence, which might be difficult. Another difficulty is that other people have actually tried mutating the set domain of these SET1-MLL proteins. And very often they find that if you mess around with the set domain, you actually affect the protein levels. So, and we have actually tried in the past, someone else in our lab had tried to make a conditional point mutant of SET1A using a conditional allele, and she found that when you did that, the levels of SET1A went down quite a lot. And so there was a really big technical challenge there.
So then another question, I guess, just to clarify, the final version of your DTAG was on the C terminus, so you also depleted that C terminal.
No, so we didn't do that specifically because when we put the tag, we did try to do that initially, and then it actually inactivated the methyltransferase domain. And so what we did was we did some a 5' race experiment to map exactly where the transcript for the alternate isoform began, and then we deduced where the start codon was, and then we N-terminally tagged the C-terminal isoform. So
Okay.
the full-length protein now actually has two diagram tags. So it's got an internal tag as well. We couldn't do it any other way.
Okay. But yeah, but so you are depleting the shorter isoform.
Yes, yeah, yeah.
Yeah. Okay. Well, one last question we have time for. Genevieve Farrell asks, did you try observing any like inducible genes or stimulus responsive genes to see how they respond in this situation?
Yeah, that's a very good question. We didn't do that. So this is all in the context of steady state transcription within asynchronous ESL cultures. We haven't tried differentiating them or doing anything to them. I don't expect there to be I don't expect responsive genes to depend on H2K4 ME3, but we haven't done that experiment.
Well, there are several other questions still in the Q&A, and Alan can answer those in the Q&A box as we transition now into our second speaker. So once again, Alan, thanks so much. That was great.
Thank you.